Hello, we've got the Generate Bio team with us today. Nemo, take it away. Why don't you introduce them and us to Generate Bio, please?
Great. Thank you, Brad. So today we are joined with Mike Nally, who is the CEO of the Generate Bio, and Jason, who is the CFO. Generate Bio is a very exciting company in the biologics, protein, therapeutic engineering. And so maybe where we can kind of start this conversation, can you kind of tell us what is the traditional kind of play when it comes to kind of biologics, antibody, or protein kind of drugs discovery?
Yeah, Nemo, thanks for the time and appreciate the opportunity. Generate was founded largely because of a frustration with how traditional protein-based biotherapeutics were discovered and developed. At its core, the way we've historically discovered protein therapeutics has largely been based on borrowing from nature. So we either cull through human plasma, or we immunize a mouse, a llama. We find something that sticks to a desired biological target of interest, and then we try and manipulate it to come up with a kind of optimal drug for human use. This is a complete trial and error process. There's a randomness to it. And in many respects, what we've tried to do from a Generate perspective is use a data-driven, more engineerable approach to revolutionize the way protein-based therapeutics are discovered and developed. And what we're seeing in the early days of the technology is certainly what we can do very, very quickly is optimize existing therapeutics to extraordinary lengths, getting to the best answer for patients very, very quickly. But we're also seeing the opportunity to create entirely new tools to prosecute biology in ways that were previously inaccessible with historical techniques. And I think that's what why the future is so exciting for us, is that we can actually drug what is considered today the undruggable and solve some of the greatest unmet needs facing humanity. Because, one of the questions we asked ourselves at the start of the company was, if you were to apply machine learning to biology, where would you start? And proteins actually became a pretty obvious choice, just given two foundational data sets that were in the public domain. One was the sequencing revolution has sequenced now every species on the planet and given us 180 million amino acid sequences. But there was this structural biology data set called the Protein Data Bank that gave us about 200,000 high quality, high resolution structures of proteins. And while 200,000 data points doesn't seem like a lot, these are actually three-dimensional data points. So each structure contains tens of thousands of data points around the 3D dynamics of a protein. And so for us, this allowed us to start to understand interrelationships between protein sequence and protein structure. But if you're in the business of therapeutics, what's actually really interesting is actually, can you add function to that equation? Because ultimately what you care about is, can you drive the desired biological effect in a human system? And so when we started Generate in 2018, the intent was actually to have a fully integrated dry and wet lab, recognizing there was a paucity of data around functional data in a well-curated manner. And so we integrate cutting-edge computation from a machine learning perspective with a wet lab that is completely focused on automating, miniaturizing, adding scale, reducing cycle time to our experimental workflows, where when we started the company, the most variants you could create to an individual target was about 100. Now we're doing sets in the millions. And that sort of data allows us to start to add that third leg of the stool, so we can find this interrelationship between DNA sequence, essentially, structure, but ultimately the biological function you're trying to drive.
There's an existing large molecule drug that plugs into a socket somewhere.
Totally.
So it addresses the condition, but maybe sorta. And so the idea is using your technology, you're able to find a better socket to plug into, or you're going after the same socket and it's like a molecule that plugs in much more cleanly, or combination of both, or it just does the same thing and it also does it in a way where it's not IP infringing, and so you can go after it safely.
A little bit of both on that one, right? So I think if you think about the history of the drug industry, let's take the statin class of drugs for cholesterol, right? You had the first statin, Mevacor, which was a monumental advance. But Lipitor, which was the third, fourth generation statin, was the disproportionate winner from a market perspective, because obviously it gave patients great efficacy and better convenience than the first generation answer. What this technology at a minimum, and what we're seeing very clearly today, allows you to do is it allows you to get to the best answer at the outset. So you're optimizing across all of the protein universe in one shot. And that allows you to basically give patients the best experience, the best answer. And if you look at the biggest drugs that have historically been around the industry, Humira was the third or fourth anti-TNF, but it ultimately was the biggest drug for rheumatoid arthritis in the world, and the largest selling drug in the industry until recently. So for us, clearly there's an opportunity to get to this best answer really, really quickly. The second thing, though, and I think this goes to your question, it's going to allow us to access biology in domains of biology that have been undruggable. Things that we know if we had the right tool to plug into that socket, we could actually drive, better outcomes for patients. We just never had that tool in our tool belt. The purpose of Generate was always not to basically predict, but to generate and create. So can we create proteins that nature has not sampled that solve a relevant therapeutic or biological task? And that's just a step further on this continuum. And the rate limiter for our ability to do that has been data. And it's these sort of data sets that we're starting to generate through cryo-EM, through functional data sets, that allow us to have a higher degree of confidence in the sequences that are actually suggested by our models, that they will translate immediately into therapeutics for benefit for humanity. The long-term vision of Generate is for every unique patient's individual condition, could we actually create a model that would give us the optimal therapeutic instantaneously?
True precision medicine. That's a precision medicine. That's a true precision medicine.
Right. And that's a long way off. I don't think we're going to unravel the complexities of biology in the next 3 years or 5 years. But I think, we know what we do know is that when we are born, what is passed between parents and child is a code. That code is DNA. That codes for all biological function. So there is an interrelationship that is to be learned. The question is how much data will it require to actually learn and master that interrelationship? And I think that's why I think over the course of the next couple of decades, this is the biologic century. We are going to start to unravel that code in massive and meaningful ways that will ultimately allow us not only to create better human therapeutics, but to solve challenges in agriculture, sustainability, material science, that these sort of technologies ultimately potentially open up.
Imagine you're trying to accelerate towards that future, and there's always like a choke point that then you're trying to efficiently throw allocation towards of resources to unchoke it, right?
Right.
What are the pinch points today and anticipated pinch points? Or maybe put it another way, with a marginal dollar, are you buying another cryo-EM machine? Are you investing in some other kind of physical testing infrastructure? Are you buying a GPU? Or are you, paying for an additional clinical trial? Maybe that's a slightly different category, but how do you think about kind of like that capital allocation puzzle to making sure you're accelerating efficiently?
Yeah, it's constantly evolving, Brett. I mean, at its core, right? And there's a couple of fronts that we actually are pushing really, really hard on. And I think you've seen this, across a number of different AI-based categories, right? The underlying machine learning methods, we have to keep pushing the frontiers on, right? And as we've seen, if you think about generative image generation, right? Over a 5-year period, we've seen from kind of grainy images with people with 6 fingers to exquisite pictures. And a lot of that's some of it's data, but a lot of it's method development, right? And we're on a similar trajectory within these machine learning models of biology to continue to push those frontiers. The nice thing about that is that's actually relatively cheap in terms of you need some world-class machine learning scientists, but once you have them, it's actually about letting them continue to push the frontiers, allow them to draw inspiration from all these other domains where we're seeing advances. to make these models more powerful in biology. On the cryo-EM side, right now, we are working kind of maniacally. We don't have a hardware problem per se today. We have enough machines. How do we make those machines more productive? How do we automate and make those machines work 24/7 and actually solve plates of structures, not individual structures at one point in time? And that to us is the biggest question. As we do that and we get to a point where those things are really, really humming, full time, then you start asking questions, do we need to add the next machine? From a wet lab experimentation perspective, a lot of what we've been solving is speed. So how do we reduce cycle times? One of the long poles in the tent has been how long it takes to grow proteins in cells. And so there are new techniques like cell-free protein synthesis that allow us to shave weeks off of those iterative cycles. So if we can grow these proteins in days versus weeks, that's a massive accelerant to everything we do. Now, each of those steps, we're constantly, as we solve one thing, as we bring in protein, cell-free, there's a new long pole.
As a close observer of, all the technologies we focus on, but really, I think we have the only, at least published forecast for cost declines in core AI that I've seen that exists, where we think performance per cost is roughly doubling every 4 to 5 months. We think that's prospectively going to happen through the end of the decade. And so, and it's clear, you can palpably feel it. The tooling is changing under people's feet in language model space all the time. And that has implications for kind of your space, for autonomous systems, for every technology we focus on. To what degree are you having to kind of rebuild the engine as you're trying to fly and come to conclusions on the plane? Do you know what I mean? How much is the kind of architectural advance within kind of the language model space flowing through into the biology space, and how does your team even evaluate or keep up with it?
We're a company that was founded in 2018, the fall of 2018. We recognize that if we don't actually fundamentally challenge the premises on which the organization was built, we will be obsolete in the not too distant future. Right? So language models were in their nascency when Generate was founded. They change how we think about science at a foundational level. Right? And, we talked a little bit about the lab in the loop type concepts, but there's no reason we're not driving agentic workflows to create an experimental loop that's fully automated and at a pace and a scale that was unimaginable. There's no reason that these large language models aren't going to be foundational in hypothesis generation. And so then running those, then testing those hypotheses in an automated setting. And then this then extends to how we run every part of our business, right? And there are pieces of this that we try and prioritize just because we can't do everything. But we were talking about clinical development being the cost bottleneck for this. There are some things here, right, where if you think about from the time a study finishes to study report closeout, right now is about a 3-month process. Why not plug a large language model on the back end and make that a 3-day process where you have all of the reports written, and then you use your talented scientists to review those reports and make the necessary modifications, right? Again, to your point, that then is adding 3 months of patent life. But we have to kind of fundamentally challenge all parts of our business on a daily basis, given this wave of technology. And we've just been really fortunate. The advantage you have as a relatively early company here, digitally native company here, is you have people who are more tech savvy in your workforce. At the same time, what we're really starting to challenge, though, is how scientists actually do their work. And, all human beings have a degree of resistance to that level of change.
But to me, this comes to a chicken and egg question, right? but because I love platforms, and that could be because I did my first internship in Flagship, so I got addicted to platforms. But you coming with that asset example, it creates value, gives you the money which you put in the platform, which then there is derivative change, and that's the flying wheel for me.
Well, that's right. And, I mean, the thing that we obsess about, and Jason and I spend a lot of time thinking about, is certainly how do we advance technology and how do we, capture, address the biggest unmet needs. But what kills biotech companies is oftentimes a capital dependency cycle, right? The ebbs and flows of science do not necessarily align with a long lead time capital dependency cycle. So if you can figure out how you balance that platform development while monetizing to reduce your reliance on investor capital. And, we think with like our anti-TSLP antibody, as it goes into phase 3 trials, now you're talking about billions of dollars of potential value for a single asset. That allows you to kind of, change this dynamic and actually really prosecute the platform thesis in a foundationally different way.
Well, we look forward to that future and kind of the incremental steps along the way that hopefully will kind of demonstrate that the platform can get these great assets to market so that you can, keep the flywheel spinning and cause it to accelerate. Thank you so much, Jason and Mike, for joining us and enjoy the conversation.
Appreciate the time today, guys.